Hi, welcome to another video by ASIJ-Tokyo iGEM. Today we are going to learn about Biobricks. Last time you embarked with us upon an extensive journey of genetic circuits. Just as a recap, genetic circuits are a system of promoters, RBS, coding regions, and terminators that promote biological processes to actually happen, such as producing specific molecules, regulating cellular behavior, or even performing computations within a cell.
Now let's get into the interesting stuff. Biobricks are DNA parts that carry out a defined function. In other words, biobricks refer to a specific fragment of DNA that is coded to perform a specific function.
There are two key components to creating biobricks: restriction enzymes and DNA ligase.
Restriction enzymes are scissor-like tiny proteins that can cut the DNA in specific places. Each type of restriction enzyme specializes in a specific short sequence of nucleotides, which are usually palindromic. This means they read the same forwards or backwards. When the restriction enzymes find their target sequence, they cut the DNA, creating a fragment. Our friend the biobrick has emerged.
There are two types of cuts that the restriction enzymes make: sticky or blunt cuts. Sticky cuts have a bit of overhang, while blunt cuts do not. These sticky cuts are helpful when you want to combine different DNA fragments together. The unpaired nucleotides on the ends make it easier for the complementary nucleotides to anneal together. On the other hand, blunt cuts are less effective due to no hydrogen bonding between the complementary nucleotides.
Once we've made the appropriate cuts, we need a way to put these parts back together. This is where the renowned ligase comes in. You might remember ligase from our DNA replication video. Just as a review, ligase is basically glue that helps connect the DNA fragments cut from restriction enzymes together. Fun fact, ends cut by the same restriction enzymes can always ligate together. Ends cut by different restriction enzymes cannot ligate unless they are blunt or their overhangs are complementary.
A lab that is widely used in the science world is the transformation lab. It is essentially the culmination of the steps we have covered. Once the desired genetic circuit is assembled in the correct order, the newly engineered plasmid will be inserted into the bacterial cells via transformation. Transformation is the process of transferring foreign DNA into a cell. In this example, we'll transform E. coli, a common bacteria found in our intestines, so that it becomes ampicillin resistant and fluorescent. To ensure that the task has been successfully completed, you will need to make sure that the bacteria glows a fluorescent green. Only then will you know that the transformation was successful. Or you can verify that the transformed bacteria is capable of expressing the desired protein. Of course, there will be some bacteria that aren't glowing due to an unsuccessful intake of the plasmid, but a majority will be ampicillin resistant if they are glowing. So, now you have a genetic circuit made up of biobricks. To summarize, first we cut out fragments of DNA, or biobricks, using our scissor-like restriction enzymes. Then we connect those biobricks with DNA ligase and create our very own genetic circuit. Finally, we check if the procedure was successful by making sure it glows or produces the correct proteins. Thank you for watching our biobrick video. Next up is PCR.